Morning. Nice to see you. Thank you so much for joining us on a Saturday morning.
Thank you so much for the invitation. Very excited to be here to Kick Cancer's KRAS.
All right, that's what we want to hear. We need to get a little tweeting going on here. I'd like to see that out there. Okay, let me do an official introduction of you since we're running about a minute behind. Okay, Dr. Awad is an MD Ph.D. is an assistant professor of medicine at Harvard Medical School, the director of the Lowe Center for Thoracic Oncology at the Dana-Farber Cancer Institute. He received his undergraduate degree from Harvard University and his MD and PhD from Johns Hopkins School of Medicine. And your translational research focuses on identifying biomarkers of response and resistance to targeted therapies, immunotherapies, and lung cancer. And he is like a regular partner with us here at the KRAS Kickers. And I'm thrilled to introduce you to Dr. Dr. Awad. Thank you so much for joining us today.
Thank you very much for the opportunity to be here. Greetings from Boston, everyone. I wish we could all be together in person, hopefully soon again someday.
Okay, and we didn't have anything like officially planned here as far as any sort of a presentation or anything. I hope you were able to join us this morning earlier for the presentation we heard from Daryl McConnell at BI about, and he kind of got like really sciencey and a little researchy. And I got to tell you, like, wow, I think I need another cup of coffee before I started the morning with it. But that's okay. Here we are now. And so
I've got one here. I wish I could pass it through the screen to you, Terry.
That's okay. I think where's your KRAS Kickers cup? Honestly,
I know. I have it in my office actually at work, and
okay. Well,
I'm calling
you need a second one. Is what I'm hearing.
I know.
All right, cool. Okay, so does anybody have any like questions for Dr. Awad immediately, or I will just like start talking to him a little bit and say and hear about what we just heard about, because it was deep, it was heavy, but you're really right out there in the throes of it with like medicine is seeing the patients. Joe McConnell does not see patients, but you do. So can you take a little bit of everything that he said and give us an actual application for us, like to a patient?
Great. I mean, I think this is such an exciting time, and I echo the enthusiasm and excitement that I think we're all feeling, that there is so much work going on in this field. You can see the size of the teams at each industry that are working on this, and here in our cancer centers and in our academic institutions, we have really large efforts devoted to studying KRAS mutant cancers. I think the pathways that were shown, the diagrams, the long lists of genes also just highlights the complexity with which KRAS can cause so many different kinds of cancers, and how difficult it is to target, and that a one-size-fits-all type of treatment won't work for every type of KRAS mutant cancer. So that's why we've seen such great successes early on with G12C, and now we're really trying to expand upon that to treat these other forms of KRAS mutations like G12V or D or A or 13 or 61. These are all the different positions along the KRAS protein that can become abnormally mutated. We number them, and each position can create sort of wreak havoc or create chaos in normal cells to cause them to start growing and dividing. So we're very excited to be partnering with our industry and pharmaceutical representatives and partners to bring some of these novel therapies as quickly as possible into the clinic.
Yeah, we as patients are quite excited too, and we appreciate your working with us and with them. Now, you previously explained to us how, like, what KRAS looks like and how it is like down a strand of pearls. Could you kind of go through that with us one more time, just so we can kind of get on the same page?
Yeah, sure. So, in a cell, there is DNA in the nucleus, and then the DNA has a code to create proteins. And a protein is made up of a string of what are called amino acids. And the string, you saw that kind of spaghetti diagram that was shown earlier. So that represents a string of amino acids, and each one has a number starting for number 1, 2, 3, 4, 5, et cetera. And so when we see these kinds of notations like G12C, that tells you that along this string of pearls, so to speak, or along the amino acids, at position 12, there was normally the amino acid called G, which stands for glycine, which again is just a particular type of amino acid there. And then the letter that comes after the number, the C, tells you that that glycine, which was part of the normal, or what you say wild type KRAS, that means the normal sequence that's used for normal cells, at that position number 12, the G became a C because of a mutation that occurred in the DNA. And that C stands for cysteine. And that is what's mutant in the cancerous cells. And that's what turns these signals on to tell the cell to grow and divide and spread and mutate further. So, so that's where we need therapies for each of these distinct types of mutations. Okay. The pan cancer, pan also means, it works against all of the KRAS forms essentially, that it's not selective or specific for one specific mutation. So the selective inhibitors, for example, the G12C drugs like sotorasib and adagrasib, really are designed to work against that specific type of mutation, but the pan KRAS inhibitors are intended to work against almost all of them or against many of the different ones. And so that's where I think there's a lot of also excitement behind these potential new drugs that stamp the protein and target it for degradation or basically just getting chewed up by the cell's machinery. So.
Okay. Well, I appreciate that. So when you, when we as patients take a pan KRAS treatment, does it respond differently in our body? I don't mean like microscopically. I mean, to me as a person, I mean, is there a difference if I'm taking an inhibitor or I'm taking a panel or anything?
Yeah, that's a great question. And I think I saw this come across in the chat. Because there is normal KRAS that normally functions in the body, and then there's the mutant form, and we again call that wild type. And then there's the mutant form that really only occurred within a cancer cell and caused the cancer cell to start growing. And so I think those are really great questions as we start to see these drugs coming into clinic really as soon as possible. It will be important to know how tolerable these drugs work, meaning if they work on not only the mutated forms of KRAS, but if they degrade or disrupt any of the normal protein, will that cause side effects or symptoms, or is there no problem with that kind of approach? And so I think that's where it's really important to work carefully with patients together who are so gracious to participate in clinical trials, because there is a lot of unknown about how these therapies may be tolerated. We need to document carefully, as patients are participating in the very earliest phases of clinical trials, any new symptoms they have, any side effects, because that's how we learn whether these drugs are safe enough to keep escalating or increasing the dose, and then also how can we combine these drugs safely for patients in clinic. So I think that's a big unknown right now, Terry, as to the tolerability of new drugs in general. Sometimes drugs can work super well and very specifically in the lab or in animals before being tested in humans. But sometimes in patients, there are unanticipated side effects that may put some speed bumps in the road for that drug's particular development. So I think that's why it's so important that we have many drug options that many companies are working on this in parallel, but also together, because sometimes something that looks really, really promising just for whatever reason doesn't work out as well for patients. And so I think it's great that we have a lot of interest in development in this space.
Okay, totally. And so the clinical trials are not something that we need to be afraid of. There's something that is really what they're working on right now, as far as the most cutting edge treatments. Is that right?
That's right. I think the drug development has been moving at such a breakneck speed, much more, rapid rates than even, 5 or 10 years ago. So I think that's really, really exciting. And I think, unfortunately, as was also highlighted, KRAS mutations in cancer are so common, and many types of cancers have these mutations. And so hopefully, because that it's such a common type of mutation, the trials will move forward as quickly as they possibly can, just because there's such a need, and so many patients are interested in new therapies. And so I think these mutations are not rare, unfortunately. And so, but because of that, I think we'll be able to learn very, very quickly about drugs and how well they work or don't work, and can move on to new things.
Okay, so what we call these are the pipeline of hope. Would you say that's a fair terminology for what's in clinic and what's being studied for right now?
Yes, 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 yeah, absolutely. I think I'm in a thoracic oncology clinic at Dana-Farber, so most of what I see is lung cancer, but I also see some patients with other tumor types. And compared to when I was in fellowship, and since I've been on the faculty for about 7 years now, it's just amazing to me to see new drugs being approved every other month in lung cancer and many other tumor types. And that was not happening even 5 or 10 years ago. So I think hope is the key word.
Okay, totally. And we call, that's where I don't know if I sent you a t-shirt or not, but it says hope shared is hope multiplied. And that's the theme of the conference right now. And that's one reason why we call it the pipeline of hope, is because that's what you guys are working on tomorrow, is what we're going to be needing. So, okay, well, I appreciate your joining us today. I lost you. There you are. Okay, so I appreciate you joining us on screen. Can we get you to do with our group like a Kick Cancers KRAS? Is
Yes,
authorized?
let's do it.
Okay, great. So here what I'm going to do, I'm going to do like at the count off. It's like a 1 and a 2 and a 3, and it'll be Kick Cancers KRAS, because it's like 3 words, right? How hard is this? Okay, so what we're going to do is, okay, one more time. This is just our way of being able to be KRAS kickers together with the people virtually, because we're just trying to make the best of a situation with this stupid COVID thing, which we are so over. I mean, it's not bad enough, but anyway, y'all have your own notions about what's going through COVID. All right, so we're gonna do 1, 2, 3. Kick
Cancers
KRAS.
KRAS.
All right, thank you so much, Dr. Awad, for joining us today.
Thank you, everyone. Nice to
Talk
see you.
later. Okay, thank you.